Our third speaker today, Dr. Armelle Pindon, is going to share some insights into the complexity in models and preclinical platforms in rare disease. Dr. Armelle Pindon is the Chief Scientific Officer of the Myhre Syndrome Foundation. She earned a PhD in neuroscience from Paris-Orsay University and has conducted research at renowned hospitals in both Paris and London, as well as having a extensive career in the pharmaceutical industry. As project and group leader in preclinical drug discovery, Armelle has honed skills in cloning, molecular pharmacology, and innovative screening technologies. In the past decade, she has dedicated herself to rare disease, supporting patient-centric organizations with drug discovery strategies. Her work includes creating preclinical platforms and tools to help develop tailored treatments and options for patient needs. So welcome, Armelle. Thank you.
Thank you. Thank you very much for having me at the iXCells. So I'm going to focus more today about multisystemic rare disease. That's I think something that is quite common to different diseases. Thank you. No, that's not the right one. Okay, so Myhre Syndrome Foundation is a patient advocacy and research organization that started in 2019 with 2 patients getting together. We are focused on the 2 aspects, community service, but also research. And recently, the research aspect has been really focused on drug discovery. So we want to have the right path and the right tools and how to choose the relevant drug discovery tools. Whatever the goal is to improve quality of life or decrease the life-threatening events or cure the disease, what we need is really to identify targets, models, and assays. So starting with the models, what everybody wants to say is, okay, we want to screen drugs and validate drugs in a model that recapitulates the disease. Well, that's not for us. Because unfortunately, we have multisystemic disease with a lot of heterogeneity. And this is a common issue with rare disease genetic disorders. So the molecular origin of Myhre syndrome is a mutation in the SMAD4 gene, which is also called co-SMAD. SMAD4 is a transcription factor integrating the TGF-beta pathways, and TGF-beta is a superfamily with 33 members and 12 types of receptors. So that's at the end of a lot of different pathways. Specifically, Myhre syndrome is missense mutation on only 2 localizations, the 500 and the 496. And the 90% of our patient is comprised in these 2 mutations, and the rest of the 10% is still at the site of the 500 with 3 others mutations. So the particularity of the mutation SMAD4 for Myhre syndrome is that it's a gain of function through an overstabilization of SMAD4 and the other SMAD complexes. So a gain of function of any kind of TGF-beta will act on a lot of different cellular and functional particularities. So from cell differentiation, and from people who do stem cells differentiation, you know that there is the SMAD dual inhibitor protocol, cell growth, cell proliferation, extracellular matrix production, angiogenesis, tissue repair, immune regulation, and apoptosis. So that's a lot. If you increase the function of all of that, that's a lot. In our case, we have to be very careful in the treatment we choose and the methodology we choose, because SMAD4 has an haploinsufficiency and the loss of function is responsible for other diseases. So we need to decrease this this gain of function and over stabilization, but we need to be careful not to lose too much of the SMAD4 activity. So just to show you the complexity of the situation, the superfamily of ligands is TGF-beta, bone morphogenic proteins, BMPs, the activins and inhibins, nodal, anti-Müllerian hormone, growth differentiation factors. So that's a lot. And you have different type of receptors which are classified in two types. The type 1 is the ALK, so the activin-like kinase receptors. And the second group is the type 2, which is the BMP, anti-Müllerian hormones, or TGF-beta receptors. So you have a dimerization and then an association between the two type of receptors. And then you have the signal transduction that we call the canonical signal transduction from TGF-beta that goes to the SMADs. So some of them will go to the SMAD2 and 3, and other will go through the SMAD1, 5, or 8. But at the end, all of them needs the SMAD4 to form a complex, enter the nucleus, and act as transcription factors. The thing is that this complex with SMAD is only a half-unit transcription factor, and you need the other half that can. We have now over 40 cofactors identified with the SMAD complex that goes from beta-catenin, TCF-LEF, with signal. So many different other signal transduction associated that we need to form the second half unit of the transcription. So you can imagine that we have a huge cell type and context dependent activity of the SMAD4. So from there, you can imagine that we have a multisystemic disease. We have neurodevelopmental deficits with intellectual disability, autism, speech delay, and nonverbal patients, some nonverbal patients, skeletal abnormalities, neuromuscular, also, it's not only skeletal, it's it's musculoskeletal, sorry. We have fibrosis, whoops, that was the wrong one. We have fibrosis and stenosis that trigger life-threatening, and in fact, the last 6 months, we had 3 deaths in kids because of tracheal stenosis and aortic stenosis. And we have widespread fibrosis in various organs, even in liver fibrosis. We have hearing loss, cardiovascular condition, respiratory condition, gastrointestinal condition, immune dysregulation, endocrine, reproductive system abnormalities, and even solid tumors. So from there, you say, well, what system am I going to take? And on what model should I focus on? But that's not all. If you find a system and you say, okay, I'm going to that system, we have heterogeneity in the system. So we have, okay, we go for neuro, we have almost 80% of seizures, but that's cumulative. You can have several of those conditions, but not all of them. Every patient is going to be different. The developmental delay seems to be something quite common. So if you go to other type of system, for skeletal or hearing, the same, you are going to see, again, a lot of heterogeneity of the phenotype that you can have. And in hearing loss, even if you have 53% of the patients that have hearing loss, we have half of them that are conductive, but we can even have some rare cases of vestibular schwannoma, like in NF2. So that's really a big issue on the model selection. Same for cardiovascular, respiratory. I'm not going to show you go through all of them, but a lot of heterogeneity. So what can you do? Where do we go? Well, we decided to go to the patient and look at the patient priorities. So through surveys, we identified that the cardiac manifestation and respiratory were the main two issues where the patients wanted to have some drugs. So I put in 4 fibrosis and stenosis because in fact the cardiac manifestation and respiratory are mainly due to fibrosis and stenosis. And the third aspect that the patient really wanted to focus on was behavioral. And if you go to the one of the patients that was mentioning what altered in adolescence was a shift in his cognition, and he developed psychosis. This is difficult to manage as a family, much more challenging than heart surgery, which he's had. So the behavior was really a big issue, even bigger than some cardiovascular. So we decided that that would be our focus: fibrosis, stenosis, and behavioral. To have the model, so we wanted targetability and modelization. The model has to have a continuity and a correlation at every level, from biochemical, cellular, organ, system, or whole organism. So the 3 questions that we ask ourselves is what is the pathway in those fibrosis and behavioral? The mechanism responsible for the phenotypes we have. Is the model sensitive to this pathway and relevant to the application? And do we have reversibility in the model we choose of the phenotype of the disease? Is it possible to have reversibility? Because we have also adults patients. So we applied some model selection criteria, which was we wanted a limited engineering intervention. So we went for the patient-derived iPSCs. We wanted feasibility and timeline, not a model that is going to take 1.5 years to get ready. Reproducibility and standardization for the screening. A good throughput, so that it won't take 6 months to test 100 compounds. We wanted an assay available or easy to further develop or validate in our models. And we wanted a relevance to the identified targets. So behavioral and fibrosis, that's still very large. We could be, so we ask ourselves, is there a coherent link between behavior and fibrosis? And in fact, we started from fibrosis. And what's fibrosis? It's wound healing that goes wrong, that goes over what it should be. So we thought, okay, that's peripheral system response to injury, that's fibrosis. What's the CNS response to injury and stress? And what's the link with the TGF-beta? So for the peripheral system, if we go for fibrosis, you have the choice of different cellular model. You have fibroblast, of course, that's the first thing you think about. But you have many other cell types, such as endothelial cells, epithelial cells, that all go through reactivity and transformation that we call the epithelial mesenchymal transformation. And this transformation leads to the creation of myofibroblasts, which is the issue in fibrosis. What about the CNS? So what's the response to injury? Well, that's not neurons. That's microglia that is modulating and responding to injury and or diseases. And the other one is astrocytes that also goes through reactivity and scar formation. So we were thinking, okay, so for the CNS, let's focus on CNS. It seems that microglia and astrocytes for our behavioral model might be more appropriate than neurons. So we checked with the TGF-beta and CNS, and we see that TGF-beta is a regulator of microglial function, inhibits phagocytosis and myelin clearance. And we thought, oh, okay, myelin clearance. In our patients, we have, in the MRIs, we have some white matter hypersensitivity, which can be a sign of demyelination and myelin issue. So that was one more point for microglia. TGF-beta is also involved in the astrocytes reactivity and synaptic plasticity. It's involved in different pathogenesis of neurodegenerative disease, but that's not our goal here. We have also a pre and postnatal macroglial cells that is regulated by SMAD4. SMAD4 is critical for driving the transcriptional signature of the microglial cells. So that was, we thought we were going in the right direction, but something that was really important for us was, what about the adults? What about reversibility of the model? Can we have a model that can show reversibility of the phenotype we have? And there is a publication on molecular diversity of the diencephalic astrocytes, reveals that adult astrogenesis is regulated by SMAD4. So that was one more point to go in that direction, but we were asking, in fact, okay, diencephalic astrocytes, are we really, do we really want us to have that? Is it relevant for our disease? And in fact, if you look at the diencephalon, which is really at the center here, you see that you have the hypothalamus, which is involved in endocrine system. And those hormones from the hypothalamus are the hormones that are involved in Myhre syndrome disease. The thalamus, where you have motor function that we have issue with in Myhre syndrome, and sleep and sensory perception. So sleep is a huge issue with Myhre syndrome patients. Again, the epithalamus is involved in the regulation of sleep, which is one of the big issues. And we have a certain amount of our patients that are already treated with melatonin, and so some improvement in the sleep. So, and the last part is the subthalamus that is involved in movement, and there are movement issue also in Myhre syndrome. So that was really to reinforce us in the astrocytes. We said, okay, astrocytes too. What about the fibrosis? So fibrosis, I told you, there's a lot of different origin of the myofibroblast. The first thing you think about is the fibroblast. Okay, let's go for fibroblast. And then you see, again, we go from a multisystemic heterogenic disease to a huge heterogeneity between fibroblast, intra and extra organ heterogeneity, and a big difference between fibroblast from human to mouse. So we said, well, could we avoid fibroblast? So we went back and looked at the different cells in the lineage of the cells, and mainly we are affected by all the different MSCs, differentiated cells. So we could go for all those different cells that would, in fact, at the end, give some fibrosis. So we went back again, asking, what about the patients? What's the burden, and what's the big issue for the patient in fibrosis? And the main one is vascular fibrosis that trigger stenosis. So we decided to go for vascular endothelial cells, and the assay will be the endothelial mesenchymal transition and wound healing assay. So we finally had our preclinical platform designed. So we sent the blood immediately to iXCells and got the iPSCs. The in vitro parts of the platform comprise the iPSCs, reprogram vascular endothelial cells, reprogram microglial cells, and organoids. And we are going to work with Alison Worthy, hopefully. So again, this name coming up for brain organoids. We have also other organoids for adapted more for fibrosis. The in vitro cellular assay we have binding assays and more biochemical assays with measuring the stabilization or destabilization of SMAD4, SMAD complex here. And then we have the functional assays from the cells, which is, as I said, fibrosis, wound healing, and glial reactivity. We hope that the astrocytes will come when we have more funding. The in vivo models are conditional knock-in mouse model. One is already available at Jackson Lab, and the other one will be on the way. The in vivo assay will be on fibrosis and stenosis, but we are starting deep phenotyping with psychogenics to really get the right screening assay specific to Myhre syndrome markers in behavioral and other characteristics. So this preclinical platform is really at the center of our pipeline, and it's really fundamental for us. So we have our treatment portfolio for now is composed of small molecule repurposing and new chemical entities, around a bit more than 100 that came from the IRB in Barcelona. Sorry, in Barcelona, the IRB Institute. They were selected, the hits were selected on the destabilization between SMAD4 and SMAD complexes. We have peptides from a company in South Korea. Our big effort is in immunocell therapy, so FAP CAR-T therapy to reduce the fibrosis. And the gene therapy editing had the first good results and is going into the iPSCs now. And the more translational research is on the neoprotein-protein interaction between the mutated SMAD4 and new cofactors, and the new brain organoid with Alison Murty and new therapy in development. And hopefully going through the preclinical platform will allow us to jump into our clinical platform that we are working on. So this preclinical platform, we hope, could be expanded to other TGF-beta related diseases, just that have fibrosis, vascular condition, neurodegeneration, or glioblastoma, or others, where you have, in fact, with Myhre syndrome cells, a kind of natural overexpression. It's not overexpression, but gain of function. So on overstabilization and over signal of all the TGF-beta. So that's, I think, a good way to screen and read any treatment that could decrease that. So that's, so I just talk about the first efforts. That's our preclinical, but we are working also on clinical readiness, and those are the treatments, our pipeline of treatment that we are working on now. And we work also, of course, on the regulatory and industry. Okay, thank you.